Okay, this is the last topic for the cell processes unit, and the topic is cell transport. Three major ideas of how cells move stuff, materials in and out of them: diffusion, osmosis, and active transport. So first, diffusion is the movement of molecules from a high concentration to a low concentration. It's just a natural process where molecules, like these little dots here, because they have heat energy, just kind of randomly bump into each other and move around. And as they do that, they end up kind of spreading out through whatever area they're in. And so that's from a high concentration to a low. Lots of things move around by the process of diffusion. For example, the smell of coffee in the air, which is a gas. At your coffee cup, the coffee's really concentrated, a high concentration, but as those fumes diffuse out through the air and the molecules bump into each other, you can eventually smell it. Diffusion also happens in liquids, food coloring. Good example we did in class where we had a flask and we dropped in some blue food coloring and it eventually spread around everywhere because of the process of diffusion. For cells, diffusion is a process they use to transport materials in and out of the cell, and it's a great choice to do it because it doesn't require any energy of the cell. The process just happens. So if you have a lot of oxygen in your bloodstream, it's going to naturally diffuse into your cells where the oxygen level is lower. So again, it doesn't require energy because the molecules have molecular motion because of the heat energy that they have. Examples of things that are transported by diffusion: oxygen, carbon dioxide, food materials, waste materials. Lots of different things can be used by this process. So the second cell transport process for the test is osmosis, which is defined as the movement of water across a cell membrane. It has to have these two things in the definition or it won't be right. So again, it's the diffusion of water across a cell membrane. So it's really, I said diffusion here, it is water moving from where there's more of it to where there's less of it, and if it crosses a cell membrane, officially it's known as osmosis. It's another example of a transport that the cell doesn't have to do anything to take in water. It doesn't require any energy. It just happens because of the heat energy, and the water always moves from where there's more of it to where there's less, a high to low concentration. One of the things that you have to be able to do for the test is understand in certain situations what will happen to a cell depending on the concentration of the solution that it's in. So calling them osmosis problems here. So if I have a cell inside of a beaker that has, looks like the little dots are supposed to be the solute, which could be salt, it could be sugar, some molecule in there, and then the empty space is the water. And so by comparison, the solution has more solutes in it than the cell itself, although the cell does have some solutes. But if you think about where there would be more water, because there's more dots on the outside, there's more water inside of the cell. And so water is going to move by osmosis from where there's more water to where there's less. So in this situation, the cell would shrivel and the water would move out. Another situation we have, I actually labeled it as percentages of salt. You think about what the percentages are. I like to turn them into water percent. Oops, messed that up. I like to turn them into water percentages, messed it up twice. It has 5% salt, that means it has 95% water outside of the cell. And if inside of the cell it has 5% solutes, which is proteins and ions and other little molecules, it's 95% water. Well, you look at them and you say, well, where's the water higher? Well, in this case, the water's not higher in either case. So what's going to happen is some water will diffuse out, some water will diffuse in, and nothing is going to really change for the cell. And that's kind of the normal situation of your cells to your bloodstream. So they don't shrink or swell. The final example here is where the cell has a lot more solute dissolved in it than the surrounding environment, I'm going to say, this time, which means there's high water here because there's less solute molecules that I drew, lower water in the cell, and that means the water is going to diffuse into the cell. That cell would likely burst, unless of the case if it was a plant cell. Plant cells have also a cell wall around them. Oops. And cell walls help prevent the cell from breaking under the pressure as the cell expands. our cells in this case actually could expand and actually burst and die. So we got a cell that dehydrates, we got a cell that bursts, the normal situation for our cells. The last type of transport is active transport. That's transport from a low to a high. It's the opposite of the other two transports, because sometimes the cell needs to move stuff to where there's already a bunch. And so this is an active process. It uses ATP, which provides the energy to actually make this happen. It always uses a protein in the cell membrane. And it can be used to move many types of molecule, many different molecules. So I have a little example here that's drawing. This line is supposed to be the cell membrane. And you could say this is the inside or outside, doesn't actually matter, but I see I have a bunch of little hydrogen ions, hydrogen atoms basically, and this is my protein here that's going to pump the hydrogen atoms across by active transport. Since I have a low concentration of them on this side, there's only one in this case, higher concentration over here, it's going to move it from here. That would be active transport because it's moving from a low to a high, and there would also have to have a little ATP molecule that binds onto this protein and causes it to pull that hydrogen across. So that's active transport from a low to a high. So those are your three transport processes overall. One more question that we briefly investigated this week is the obvious question of cells are so small, and we were wondering, well, why is that? And here's a true fact that our math calculations of the lab pointed out. It says small cells have a larger surface area compared to their volumes. And what that ends up meaning is that smaller cells end up, and we have two pictures here, they can allow in a lot of materials, and that helps them survive. But the main point that we focused on was that smaller cells, the time it takes for something to diffuse all the way across or to the center of the cell, say it's oxygen, is a lot shorter because the length of this arrow represents time. The same amount of time in a larger cell, an oxygen molecule could only diffuse that far, meaning all this area here would not have enough oxygen to do its cell respiration and make ATP. It wouldn't be able to survive. Smaller cells, on the other hand, have a much easier time getting diffusion to happen all the way through them in a timely fashion, which allows them to survive. Which is why you don't see any big cells, because the smaller cells have this easier way to survive. So I can't remember exactly what I was going to write here, but I think I just said it. It says cell transport processes are most efficient when the cell is small. So bottom line, cells are small because the transport, diffusion through the cell, transport of materials in the cell can happen a lot timelier. It's not that they happen faster. It's just that there's a shorter distance for the stuff to diffuse in a small cell than there is the distance to diffuse in a large cell. Shorter distance means you can get everything you need wherever you want fast enough, and you're going to be able to survive. And that goes for waste too. If you need to get rid of carbon dioxide or other wastes, if you're small, you have an easier time getting it out. So that's the key. That's the end of the unit. Good luck on the test.